big hello to my wonderful PU2 champions. So finally we have a biology preparatory examination, and I know there is not too much time, which is why I'll not take away any bit of your seconds out there, my dear children. Just one very important thing. This video will help you with all the most important chapters out there. There are certain other chapters which I'm not covering because I know it will be overburden for you if at all I come up with everything all together. But yes, you have the PDF. Please refer whenever you go through bus or anything like that. You can just refer to the answers which are given in this particular PDF. Wishing you all the best, and the speed might be way too much. If at all you want to lessen the speed, go to the settings and lessen it up. Wishing you all the best, lots and lots of love. Let's go ahead. Going ahead with the first chapter that we have, and that is sexual reproduction in flowering plants. And the first is the very important question: what is the significance of sporopollen? Sporopollen is a very important and the most resistant organic material out there. It forms the exine layer of the pollen grain, and my dear children, it can resist alkali, acids, any pH, which is why it can be stored as pollen grain can be stored as what? Fossils as well. Always remember, this is part of exine. Inside that you have intine. Intine is made up of Priyanka Chopra, P for pectin, C for cellulose. Inside that you have plasma membrane, and inside that you have two very important cells. One is the very big cell, which is known as vegetative cell, and inside that floating, what do you have? You have the generative cell. Vegetative cell has an irregular nucleus. Generative cell has a spindle shape or elongated nucleus. Elongated nucleus would be more right, so write that. Now the generative cell can divide into two to form two male gametes out there, and so in that particular case, the pollen grain will be having 3 cells, otherwise it just have 2 cells. So 60% of the angiosperms, that is majority of the angiosperms, release the pollen grains in the 2 cell stage. 40% of them which are left are going to release the pollen grains in 3 cell stage. I hope that is crystal clear. Now going ahead, micropyle remains a small pore in the seed coat of the of the seed. Why so? So basically, if at all you see the structure of the ovule, my dear children, you have 2 integuments, and this is your ovule, and here you have the micropyle region. Why do you think it remains like that? Why do you think it remains like a bore? Because you need to make sure that once the ovule transforms into a seed, it gets enough amount of moisture, air and everything out there, which is why the micropyle is still there. Now apple and cashew are not called true fruits. Why are they not called true fruits? Because usually what happens is that this ovary which is there outside the ovule, it is the one that transforms into fruit. But in some plants, some other parts of the flowers also contribute to what? Fruit formation, and that is known as false fruit. Apple and cashew are examples of what? False fruits. So micropyle remains as a small pore in the seed coat of the seed. Why? Because, my dear children, it helps with the entry of water and oxygen during what? Seed germination, which is very, very important. Apple and cashew are not called true fruits. Why? True fruits develops only from ovary after fertilization, but over here they are developing from other floral parts also. So if at all you see the structure of this particular apple, here you have mesocarp, endocarp and everything, which is the part of the ovary. So ovary is definitely taking participation in formation of fruit, but the majority of the part is from thalamus. Thalamus is another floral part which is helping in the formation of fruit over here, which is why you call this as false fruit. Always remember that. Now differentiate between pericarp and perisperm. So the walls of the ovary that we have is known as pericarp. How many layers do you have? You have endocarp, mesocarp and exocarp. Exocarp is the outermost layer that we peel off. Mesocarp is the thing that we eat, and basically surrounding your seed you also have one more layer, the thick layer that we have, it is known as endocarp. Always remember. Now whereas when I talk about perisperm, what is perisperm? So my dear children, if at all you have seen the structure of ovule, in the structure of ovule you have cells around over here, you call it as nucellus. Initially that nucellus is there, later on it usually disintegrates or basically goes away. But over here in some, wait just a second, in some of the plants out there, this nucellus still persists. And when in the ovule the embryo is being formed, the nucellus is going to provide nourishment to the embryo. Along with the endosperm and everything out there, this nucellus also helps in the nourishment of the embryo. That is known as perisperm. Only in 2 plants, B square, that is beet as well as black pepper, you will be finding perisperm. So pericarp is the outer layer of the ovary, perisperm is something else. Going ahead, difference between albuminous and non-albuminous seeds. Albuminous seeds basically means what? The one where the endosperm is still persistent. Listen, after double fertilization, 2 things happen. One is basically you get the zygote. The second thing is you get the endosperm. The endosperm is going to nourish the zygote to form the embryo. Sometimes the endosperm is used up in plants. It's used up in plants. How? Because when the zygote turns into an embryo, the embryo says that I can store my own food in the form of cotyledons. I do not need endosperm anymore. So the endosperm, whenever it is used used up. Okay, it is known as non-albuminous seeds, and here you will be finding embryo with 2 cotyledon out there. So whenever you can break the seed into 2 parts, just understand they are dicots. Example, bean, pea, groundnut, all of them I can break into 2. That means they have cotyledons instead of what? Endosperm. So they are considered to be non-albuminous seeds who have completely used up their endosperm during the embryo development. On the other hand, there are some plants which still have some endosperm left. Rest of the endosperm is utilized for formation of only 1 cotyledon. You call them as monocotyledons. You also say that they have what? Albuminous seeds, because some amount of endosperm is still persisting. For example, the things that you cannot break into 2. For example, wheat, maize, barley, castor, coconut, everything. Coconut you can break into 2 like this, but if at all you see the embryo, my dear children, you cannot break into 2. Okay, so this is albuminous and non-albuminous seeds. Now draw a labeled structure of two-celled pollen grains. Please remember, you have to draw everything which is over here. Super important. Okay, I have explained the structure of pollen grains. Now, LS of an embryo of a grass, this is also super important. Always remember, you have the plumule over here. Covering the plumule, you have the coleoptile. Before that, radical, root cap, and then here you have the coleorhiza. One scutellum is there, and that has been mentioned over here. The other part is epiblast. If at all you are asked about the dicot embryo, please do not worry. Just make the diagram like this. This is going to be your plumule. This is going to be your radical. This is going to be your root cap. And now from here, you need one cotyledon, which goes like this, and another cotyledon that goes like this. And your picture is complete. This region below is known as hypocotyl. This region is known as epicotyl. At the tip of it, you have plumule. Remember, over here, covering the plumule, you have coleoptile. Covering the root cap, you have what? Coleorhiza. So that is the only difference. Coleorhiza has an R, radical also has an R, coleoptile has a P, plumule also have a P. Over here, the cotyledon's name is also given since it is only one. It is known as scutellum. It is known as scutellum. Always remember, this is the embryo of a grass, and grass is a monocot. Grass is what? A monocot. Going ahead with the next thing, draw a neatly labeled diagram of typical anatropous ovule as an ulta ovule. So basically in ovule, what is there? You have 2 integuments like this, and then my dear children, it's inside the ovary. Inside the ovary, you have the placenta. Placenta is attached to the ovule with the help of a structure that is known as funicle, and the point of attachment is known as hilum. Now suppose this whole thing is shifted this side. Then you have the placenta over there, funicle over here, and hilum over here. This area where the integuments are not there, you call it as micropylar region, and this area where the opposite of micropyle, wherever it is known as chalaza. It is known as chalazal end, my dear children. Now the funicle can be here, still the chalazal end will be here itself because the micropyle will be here. So the opposite end is only considered to be what? Chalazal end. I hope that's clear. Anatropous ovule means ulta ovule, where this particular part is given over here, and I gave you the picture of this. Inside the ovule, you have the nucellus also, the cells of the nucellus. One of them is going to become the megaspore mother cell and going to undergo the meiotic division to undergo megasporogenesis. Only one functional megaspore is there, and this functional megaspore is going to form the entire embryo sac with the help of 3 mitotic division. Now, why are some seeds referred to as apomictic seeds, and mention its advantages to a farmer who uses it? So why are some seeds known as apomictic seeds, my dear children? So usually what happens, you have the female gamete, and the female gamete fuses with the male gamete to form a diploid zygote. But over here, the gamete is already diploid in nature. So basically, this particular seed will only able to, this particular plant which is there, it already has a diploid female gamete and it will form the zygote. So you do not need fertilization to happen. So now the plants which are going to be formed will be looking exactly like this female parent plant out there. And suppose this female parent plant is super good, it is pest resistant, this and that, every feature of it will be transferred to the seedling or the new plant out there. This is also termed as asexual mode of reproduction. It is usually seen in grasses and Asteraceae. It is seen in grasses and Asteraceae. It is genetically identical to the parent plant, which is exactly why farmers use them. Why? Suppose a farmer is having a good plant and now it wants to create clones of this particular plant. Then in that particular case, the hybrid nature is being maintained, which is why you can choose the apomictic seeds. Also, it is cost effective. You do not need to buy every time hybrid seeds out there. One plant is enough. If at all it is undergoing apomixis and producing apomictic seeds, you do not need to buy hybrid seeds every single time. And uniformity in the groups. All the crops, my dear children, are genetically going to be uniform as well. These are the reasons. Please go through them as well. Now, example, always remember grasses and Asteraceae. One more thing, a polyembryony is also given. Polyembryony means same thing. Over here, if at all you are forming multiple embryos within the, usually only one embryo is being formed, but think that multiple embryos are being formed. Here also fertilization is not required. Suppose the nucellus is still persisting. I told you in some cases persist, right? Some cells of the nucellus gets inside and forms embryo. In this case, you call it as polyembryony. You see this in citrus fruits. Citrus fruits as in orange, orange and this thing. What do you call? Mango, yeah, orange, mango and things like that. Now, plan an experiment and prepare a flowchart of steps that you would follow to ensure the seeds are farmed only with the desired set of pollen grains. So basically, if at all there's a crop improvement program and you want to produce hybrid variety of seeds, what do? You do hybrid variety of plants out there. What do? You do artificial hybridization, which looks like this, my dear children. So suppose a plant has two things, that is the male and the female part. You do not need the male part, otherwise the pollen grains will land on the stigma. So first thing that you do is emasculation. You remove the male part, emasculation. You remove the male part. Now what do? Basically you cover this particular plant up so that no other pollen grains can come and line until it becomes receptive. Now the moment it becomes receptive, what we do? The pollen grains that we want, we will put it over this and we will rebag it. We will do what? We will rebag it. Okay, there is bagging and rebagging also, and then we will wait for the entire process to happen. So over here the entire process is given down. I'm not reading out everything because the time is not up there. Okay, but yes, you were there in the live. If at all you were not there in the live, please download the PDF. You can read through if at all you're going through bus and things like that. Okay, but don't worry. And also one more thing, if at all the flower is not bisexual, that means it does not have male and female parts, then you need not do emasculation. Okay, you need not do emasculation. Now, importance of such experiment, they prevent contamination by unwanted pollen and help in producing hybrids with desired character. Now, this question can be asked in different ways, that a farmer wants to create a hybrid variety of plants. What crop improvement program will he use? Crop improvement program comes, then understand it is artificial hybridization that is being asked about. Now, picture related to pollination is given, that means insect pollination is given over here. You have to talk about, will this pollination confirm fertilization? Do you think pollination confirms fertilization? No. Pollination means landing on the stigma. Fertilization means fusion of male and female gamete. So pollen grains can land on the stigma. They can either also form the pollen tube, but not all pollen tubes comes and reach there. And sometimes they are not. The pollen tube, the pollen interaction, the pollen pistil interaction also does not take place, which is why fertilization need not happen, my dear children. Does the plant give some floral rewards to the insects? Yes, it gives nectar, it needs pollen grains, it also provides what? Shelter as well. Now, characteristics of flowers in insect pollination. Flowers basically are large, colorful and fragrant. They are sticky or spiny pollen grains so that they can stick on the surface of the insect, and the stigma is sticky to capture the pollen grains brought by the insect. The foul smelling flowers are there in plants, pollinated by flies and beetles. Similarly, remember the examples of wind and water pollinated plant. Wind pollinated, you have grasses and corncob. Water pollinated, you have Vallisneria as well as this thing Zostera. Remember, water lily, water lotus, even though they are in water, they do not undergo water pollination. Okay, now complete the given tabular column with respect to the male and female gametophyte of angiosperms for the mentioned character. Number of cells in the male gametophyte. What is the male gametophyte? It is the pollen grain. The one that forms the male gamete is the male gametophyte. What is the female gametophyte? It is the embryo sac. So my dear children, the number of cells present in the male gametophyte is what? Either 2 or 3. Okay, either 2 or 3. If at all you see the structure. Okay, I'll just say. So this is vegetative cell, and then generative cell. The generative cell can divide into 2 also. So 2 or 3. If at all you talk about the female gamete, over here you have 3 antipodal cells, you have 2 synergid cells, 1 egg cell, 3 of them together comes and forms egg apparatus. And then you have 1 big cell which is known as central cell. The central cell has 2 polar nuclei. So 1, 2, 3, 4, 5, 6 and 7 cells is there, but nucleus, how many is there? 1 extra nucleus is there, so you call 8 nucleate 7 celled condition. Okay, a 7 celled 8 nucleate condition up over here. Always remember that. Now if at all you solve this, okay, just a second. Now if at all you try to solve this, so you get number of cells present in the male gametophyte 2, 1 vegetative, 1 generative, or 3, 1 vegetative and 2 male gametes. Over here you have 7 cells. The name of the 7 cells have been mentioned. Number of gametes present in, there are 2 gametes present in the male gametophyte, whereas there is only 1 in the case of mature female gametophyte. Type of cell division involved in the development of the male gametophyte. To form the male gametophyte, you need to undergo mitosis. In the case of embryo sac also, you need to undergo mitosis. So over here, the microspore from the microspore which was there, that forms the male gametophyte. When it forms the male gametophyte and undergoes mitosis to form the 2 cells out there, then again mitosis to form the gametes out there. Similarly, here 3 rounds of mitosis happens in the case of female gametophyte. Please remember it. Now, the ploidy of cells, they are all haploid. They are all going to be haploid in the case of male gametophyte. In the case of female gametophyte, my dear children, not all cells are haploid. Why? This haploid. But central cell, it has 2 nuclei. 1 nuclei haploid, another nuclei haploid, diploid in nature. Since it has 2 nuclei, central cell is going to be diploid in nature already. Okay? So please remember that as well. Now, what is there? Advantages of seed. Seed provides what? Protection and dormancy. Protection and dormancy, my dear children, because seed coat is there. Remember the integuments which are there in the ovule, they transform into what? Seed coat, testa and tegmen. And they provide protection and also dormancy. Food for the embryo. The seed has endosperm, my dear children, which is why endosperm or cotyledon, which is why both of them are going to provide nutrition. Dispersal also. Seeds are super important for dispersing by wind, water and everything out there. Cool? Cool. Now, next thing, explain the different outbreeding devices by the flowering plants. So, one is outbreeding means you do not want to undergo pollination, you want to undergo cross pollination. Those are outbreeding devices. So, first thing is time difference. Pollen release and stigma receptivity is not synchronized. Second, spatial difference. That means anther is over here, stigma is over here. Pollination will not be happening. Self incompatibility. This is genetic mechanism. But even the pollen grains land, the pollen postal interaction will not be taking place properly. Now, the next one is unisexual flowers. Now, there are some plants which are unisexual. Plants, as in one female plant, one male plant. In that, obviously, what is going to happen? Cross pollination is going to happen because only female plants are over here and male plants are over here. That is dioecious plants. Example is papaya. You call them as dioecious, where the sexes are separate in two different plants. For example, papaya, female plant is there, male plant will also be there. There are monoecious plants such as castor and mace also, where similar things happen, where male and female flowers are there in the same plant, but male and female flowers separately. So over here also, to an extent, cross pollination is going to happen, but geitonogamy can also happen. Geitonogamy can also happen up over here. Pollen from the one flower to the another flower of the same plant. That can also happen. Okay, so please remember that. So it is not true cross pollination, but yes, to some extent it can prevent it. Okay, double fertilization is a unique event arranged. Okay, double fertilization is a unique event. I hope you know that one particular thing which is there. Okay, one particular male gamete fuses with the female gamete to form the zygote. The other male gamete fuses with the two polar nuclei. So male gamete has a nuclei. These two nuclei are also there. So there are three nucleus which are fusing, which is why you call it triple fusion. And the end product is endosperm, where you have triploid nature. Zygote is 2n and that is 3n. So this is known as syngamy. Okay, this fusion is known as syngamy and that is known as triple fusion. I hope that is clear. And now we go to the next chapter, that is human reproduction. My dear children, obviously there might be some other topics as well, but this video intends to just help you with the last minute revision at least, because obviously at last minute you cannot revise everything, right? So this is it. Going ahead with the other things, that is human reproduction. The first thing is what is menstrual cycle. Mention one pituitary and one ovarian cycle, ovarian thing. So see, this menstrual cycle questions would be very easy for you if at all you look at this picture very carefully. So from the 1st to 5th day, you have the menstrual cycle, menstrual phase. Okay, what phase? Menstrual phase. In menstrual phase, the level of estrogen and progesterone is going down. The level of estrogen and progesterone goes down. And when these two guys goes down, my dear children, the pituitary hormones, you have two pituitary hormones and two ovarian hormones. These are the ovarian hormones, these are the pituitary hormones. FSH and LH goes up. FSH and LH slowly goes up, and my dear children, in this particular period, slowly after that, the follicles are going to develop. Follicles are going to develop. Now, when these follicles are developing under the influence of FSH and LH, one very important thing happens. The granulosa cells of these follicles are going to release some estrogen, which is why the level of estrogen is going to up. Now, menses happened. Now, slowly endometrium is going to regenerate under the influence of what? Estrogen. So, which is why you call it proliferatory phase, because this thing is proliferating, and you also call it follicular phase because follicles are also developing. Now, what happens is that when the level of estrogen goes really high, it causes LH and FSH surge, especially LH surge. At this particular point of time, the ovulation happens. The mature follicle breaks open and releases the secondary oocyte. This is known as ovulation. This happens in the mid of the cycle. Suppose the cycle is of 28 days. On 14th day, you will be having ovulation. Okay. Now, my dear children, whatever is left behind in the follicle, you call this as corpus luteum. And this corpus luteum is staying because LH and FSH is there, especially LH. Now, corpus luteum, my dear children, will be releasing some hormone, that is progesterone, and also estrogen, which is why the level of progesterone goes really high. If the level of progesterone goes high, my dear children, endometrium also thickens very much, because this is a very important hormone out there. And also estrogen is also there. This phase is known as secretory phase, because now the endometrium which is there, it is starting to secrete some very important things, thinking that embryo will be coming and basically I have to nourish it. But if at all that happens, it happens, otherwise not. So it is also known as luteal phase. Why? Because corpus luteum is doing everything out there. Now, what usually happens is that progesterone, when its level is very much, it does not allow FSH and LH to go up. It makes it drop. And when they drop, my dear children, corpus luteum will not be maintained, which is why the progesterone will also go down and estrogen will also go down, and again you have the menstrual cycle happening up there. Now the question is, what are the functions of the hormones in the menstrual cycle? FSH is a pituitary hormone. It stimulates the growth and maturation of ovarian follicles and also induces the follicles to release what? Estrogen. Estrogen, what does it do? It basically repairs and proliferates the uterine endometrium and also leads to LH surge, causing what? Ovulation. These are the important things that you have to keep in mind. Now what is placenta? List its function out there. So placenta is something that is being formed partially by the embryo and partially by the uterine tissue that is there. Okay, mother's tissue out there. So in the embryo which is going to come and settle up over here and form the uterus, it is known as blastocyst embryo. Blastocyst stage of embryo, where the outside layer is known as trophoblast, and my dear children, the inside layer is known as inner cell mass. Inner cell mass. So when this comes and attaches on the endometrium, it forms finger-like projections like this. You call this as chorionic villi. And from the mother side also, you have some maternal tissue rounding up. Together now this forms a thing that is known as placenta. Now this inner cell mass which is there is going to form the actual embryo out there. Okay, going to form the actual embryo out there. So placenta is a vascular connection between the mother and the fetus that helps in nutrition, respiration, excretion and hormone secretion. How does it do that? It's written over here. Respiration as in supplies oxygen, removes the carbon dioxide. Similarly, excretion removes all the metabolic waste out there. And also it's a temporary endocrine gland. Why? Because it releases certain very important hormones such as what? HCG, human chorionic gonadotropin, HPL, progesterone, estrogen. There are 4 hormones released by placenta. Please remember, only 4 of them are there. Okay, only 4 of them are there. During pregnancy, there is an increase of 3 other hormones also. Cortisol, then what do you have? Cortisol, prolactin and thyroxine. Cortisol, prolactin, thyroxine, their level also increases in multiple folds in the blood during pregnancy. They are also already there, they increase their production. Okay, now ovary which is there, it also releases one hormone towards the end of pregnancy, and that is known as relaxin, to relax the ligaments so that the baby can easily move out. That is also one very important thing. Remember, there are 4 hormones, only 4 hormones. Okay, now name the pituitary and testicular hormones involved in spermatogenesis. So spermatogenesis, my dear children, you know, the primary spermatocytes which is there, they divide to secondary spermatocyte. Again, they form the spermatids out there. Spermatids, when they are found, my dear children, they are immature structure. They are what? Immature structure. Okay, spermatids are immature structure. You need to make it mature. And this process of converting the spermatids, which are globular structure, okay, they are globular structure, to spermatozoa, which is what having tail and everything. This process is known as spermiogenesis. Spermiogenesis. And this can only happen if at all FSH is there. Why? Because FSH which is there, it tells the Sertoli cells. It tells the Sertoli cells to release some factors. Now these factors make sure that the testosterone which comes into the testis, my dear children, it stays over here and helps in the whole process to happen. That is why it is super important. So LH which is there, okay, there are 2 hormones, pituitary hormones. One is LH and the other one is FSH. LH is going to act on the Leydig cells, and the Leydig cells are going to produce what? Testosterone. Okay, FSH is going to act on Sertoli cells, and Sertoli cells are going to produce what? Factors. And my dear children, over here spermatogenesis is happening, so you want the testosterone to be present in the entire procedure. So that's why you need to release some factors so that this testosterone maintain its concentration, especially during the last stage of spermatogenesis, which is known as spermiogenesis, where the immature globular kids, spermatids, mature into spermatozoa. So the most important thing that usually happens, if at all FSH is not there, this conversion is the one that is going to get hampered. Okay, yes. Now, parturition is induced by complex neuroendocrine mechanism. Comment. Parturition, my dear children, starts off. Parturition means expulsion of the baby outside the womb. It starts off through the baby itself. The baby, the fetus, fully grown fetus and the placenta, they give something that is known as fetal ejection reflex. So baby which is there, it tries to move around. When it tries to move down, it activates all the stretch receptors out there in the cervical region, which is why, my dear children, a whole loop of nervous system gets activated. Apart from that, the placenta also releases certain hormones up there. Now these hormones are going to make some changes in the uterus. So both the baby as well as the uterus, as well as the placenta are going to do the fetal ejection reflex. And now when the baby moves down, a nervous system, the neuronal signal is being activated, and this neuronal signal goes to the brain of the mother. It goes to the pituitary, posterior pituitary of the mother, and the mother will now release a hormone. So nervous signal transforming into a hormonal thing, it releases oxytocin. Oxytocin will now come and bind on the receptors on the myometrium, the muscular layer of the uterus. Okay, and it will cause contraction relaxation. This is a thing. Now, draw a label diagrammatic view of female reproductive system. This is how it looks. Make sure you draw it properly, my dear children. Draw a neatly labeled diagram of sectional view of mammary glands. All the important labelings are be mentioned over here. Please draw it. Please look through the picture at least one. Write the schematic representation of spermatogenesis. So from here, you need to understand the primary spermatocytes are there. Then you have the secondary. Why is not zooming up? Okay, primary spermatocyte undergoes meiotic division, secondary spermatocyte, then you have spermatids. It undergoes differentiation or basically spermiogenesis to form spermatozoa. The release of spermatozoa from the seminiferous tubule is known as spermiation, aviation. Please remember that also. So until here it was diploid, 46 number of chromosomes was there. But after the first meiotic division, it becomes haploid in nature. Also remember one very important thing, in spermatogenesis you get 4 sperms. But in oogenesis you get only 1 ovum at the end. Only 1 ovum at the end. Now draw a neatly labeled diagram of the human reproductive male reproductive system, and over here I have mentioned everything. Please go through it. Important other topics, hormones involved in gestation and parturition, and also the months which are there. So most of the questions I did in MCQs, which is why I'm not able to discuss over here as well. But I'm telling you, please remember the months. So in first month, heart is being formed. Second month, you have limbs and digits being formed. In the end of the first trimester, major organ and organ system. In the 5th month, hair. So I'll tell you the easy way. When we die, the first thing that we check is heartbeat. So here also the first month, the heart is the first thing that is being formed. In the second month, you have limb and you have digits. In the 3rd month, you have some organ systems being formed, and also you have the external genital organ, male or female. You will get to understand that by the end of 3rd month, or you call first trimester. Now each month has 4 weeks, so 3 into 4 is 12 weeks. By the end of 12 weeks, all of this thing has taken place. Now 4th month skip, 5th month baby has God please make me move. So movement, first movements, 5th month first movements, and also first hair on the head of the baby comes in the 5th month. Now 6th month, God says you wanted hair, no, have hair on the body also. So there will be hair on the body, there will be eyelashes, eyelids will be separated. Everything happens in the 6th month. Okay, that is end of the 2nd trimester. Okay, now last month is given 9th month, and the fully developed baby is going to move out. Now in the case of male accessory glands, there are a few things to remember. Remember, there are 3 glands: seminal vesicle, prostate gland, and bulbourethral. Seminal vesicle and bulbourethral are paired structures. This is an unpaired gland. Seminal vesicle releases what? Fructose. Fructose is very important for enrichment of sperm to provide energy to the sperm. Prostate on the other hand gives calcium and enzymes to the seminal plasma, and bulbourethral glands helps in the lubrication. Remember, all of the secretion together you call it a seminal plasma. It has calcium, it has fructose, it has enzyme. Calcium and the enzymes are released by prostate, and fructose is released by seminal vesicle. Okay, done. Now you have the chapter reproductive health, my dear children, and in reproductive health, what do you have? Mention any 4 assisted reproductive techniques that be advised to couples with infertility problems. You have IVF, in vitro fertilization, okay, where fertilization happens outside the body in the laboratory, my dear children. Later, this particular embryo or the zygote which is being formed can be transferred to the body. If at all you are transferring the zygote, okay, and the zygote, or if at all you are transferring the embryo which is up to 8 blastomere stage, this is known as ZIFT, zygote intrafallopian transfer. If at all, my dear children, you are basically transferring an embryo more than 8 cell stage, you are putting directly in the uterus, you call this intrauterine transfer. Okay, now gift is also there when basically suppose a female is not able to produce ovum, rest she is able to provide all the important atmosphere up there. So you can take gift as an ovum from a donor, and this ovum can be put in the fallopian tube, and natural fertilization will be happening inside the body, not outside the body. This is in vivo fertilization that happens in the gift. And artificial insemination is there when the semen is not being produced by the donor for some reasons, or probably the semen is produced but the sperm does not have motility. In that case, artificial insemination is done in the case of males. Okay, so these are the 4 reproductive techniques that can be used. There are more than 4 I am given over here. Now mention the reasons for infertility in humans. It can be physical reason, okay, such as structural defects in the reproductive organs, congenital reasons, as in something that is from birth. Okay, for example, my teacher, hormonal imbalance and things like that. Now disease related causes, for example, you have some STIs and things like that, which is why infection is there and disease is there in the body, which is why proper production is not happening. Now drug related can be there. Certain drugs, my dear children, can lead to infertility issues. It can be immunological, where your own body can kill your own cells out there, or it can be psychological as well, where emotional factors may lead to infertilities. These are the reasons for infertility in the case of human beings. Now mention any 2 events that are inhibited by the intake of oral contraceptive pills to prevent pregnancy in human beings. What happens when someone takes oral contraceptive pills? First of all, please know oral contraceptive pills have 2 major enzymes. One is progesterone, another one is estrogen. Okay, progesterone and estrogen, they are found normally in our body, but if at all their amount is being disturbed by us, it will cause what? Trouble. it is going to cause trouble. It is going to cause something which is usually not doing. For example, progesterone is the one that helps in, estrogen is the one that helps in ovulation, but over here ovulation is not going to happen. Progesterone is the one that helps in implantation. That means the blastocyst comes and implants into the endometrium. That is also not going to happen. Why? Because of the same reason, because their level is not maintained now. So ovulation and implantation is two major things that are inhibited by oral contraceptive pills or wherever the hormones are there. For example, if at all you're talking about implants, if at all you're talking about emergency contraceptives, if at all you're talking about hormone releasing intrauterine devices, everywhere these are the problems that are being caused by them. Now, what are emergency contraceptives? Right there, hormonal combination. Emergency contraceptives are something that you give in emergency situation, such as between the 72 hours of coitus, you should be giving it to them so that conception does not happen. Either you can give progesterone or progesterone estrogen combination pills, or you can put hormone releasing IUDs out there. Okay, these are all important for preventing pregnancy, and these are emergency contraceptives because they prevent ovulation, and ovulation, if at all ovulation has happened, they prevent implantation at least. Okay, now write any 3 principles to be followed by people to be free from sexually transmitted diseases. First of all, avoid multiple partners. Second, current and consistent use of condoms, because condom is the only contraceptive which it obviously prevents conception but also prevents sexually transmitted diseases. Now prompt medical checkup and treatment, and avoiding sharing needles and razors, where, my dear children, certain STIs are not just transmitted through the vaginal or the other fluids out there, the sexual fluids, genital fluids. Apart from that, it can also be passed through bloods. So in that case, please avoid sharing needles as well as razors. But these are the 3 points that is written in your NCERT first, so please write them first. Now, define venereal disease or sexually transmitted infections. Venereal or sexually transmitted infections are the ones that are transmitted primarily through sexual contact from an infected person. Now, modes of transmission: unprotected sexual intercourse with an infected person, sharing contaminated needles or syringes. Example, drug abuse. In the case of STIs, that can be transmitted through blood. For example, HIV and things like that. Transmission of infected blood or blood products, or also from infected mother to the fetus. That can also be happening. All of this is basically where the STIs are caused through blood also. The other fluids also. So there are 3 STIs which cannot be cured. All of them starts with H. Hepatitis B, herpes, genital herpes actually, but herpes is there, and then HIV/AIDS. Okay, they are not curable. Now, what is medical termination of pregnancy? Mention any 2 medical grounds on which pregnancy can be terminated. So, my dear children, what is medical termination of pregnancy? It is intentional or legal termination of pregnancy. Okay, and it is induced abortion, you also call it. And now, when can people do that? Once, when it is risk to the mother's life, or when there are some severe genetic abnormalities in the baby. In that particular condition, we can do it. And my dear children, always remember the time it was legalized is 1971, and the Medical Termination Amendment Act was put in 2017. So 71 ulta 17, you can remember like that. So 1971 and 2017, one where it was legalized and the other one where basically it was Medical Termination Amendment Act was being put. Okay, and usually MTPs are performed after amniocentesis. This is also very important. Amniocentesis means basically the baby fetus which is there, it is covered up by amniotic fluid. If at all you take the fluid, you will also get some shed of cells of the baby. Now you get cells. Now cell has the nucleus. Nucleus will be having the DNA. With the help of the chromosome structure and everything out there, you can find the sex of the child. But amniocentesis was not introduced to find the sex of the child. It was used to know about the genetic disorders, if anything is there in the child. Genetic disorders such as thalassemia, such as Down syndrome and things like that. I'm not talking about jaundice. Jaundice is not a genetic disorder. You cannot find it through amniocentesis. Please remember that. Okay. Okay, now the popular and the effective contraceptives includes IUDs, intrauterine devices. Mention the types of IUDs with an example of each. Non-medicated IUD. Non-medicated means they are not going to release anything. They are just going to be there in the body. And since they are there, our immune system becomes active because some foreign thing is there. So anything foreign that comes up along with it will also be killed. For example, sperm comes up, it will be killed. Example is lipase loop. It looks like a loop and it is put inside the uterus. Now copper releasing IUDs such as CUT and copper 7. Please remember their name. CUT, copper 7, and also multi-load 365, multi-load 375, yes, multi-load 375, and also copper T and also copper 7. They are also super important. They are all copper releasing. What do they do? Copper ions, my dear children, does not allow sperm to move, and if at all sperms are moving, they do not allow fertilization to happen properly. That is the thing. Motility and basically the viability of the sperm is being lost out there. It prevents fertilization. Hormone releasing ISD, example is LNG20. So I remember as CNG20. So my dear children, don't remember like that. LNG, levonorgestrel, it is the other form of synthetic progesterone. progesterone. Similarly, progestasert is also another form of synthetic progesterone, and this can be given to cause a hormonal imbalance that can lead to what? That can lead to stoppage of ovulation, hindrance in implantation, and also the cervical mucus, my dear children, becomes very thick. It looks like a plug, so sperm cannot enter through the cervix. That is also there. Now, and also remember one very important thing, IUD is the most widely accepted contraceptives along with oral pills. Oral pills are also widely accepted, and if at all, if someone is looking for spacing between two children, IUD is the best. IUD is considered to be best. So two widely accepted contraceptives are what? IUDs and also oral pills. In that, IUDs are there for spacing the children as well. Now, what is contraception? Briefly explain two natural methods and two surgical methods. Natural method is periodic abstinence, where there is no sexual intercourse between the 10 to 17 day. Please remember the day 10 to 17. Why? Because this is a fertile window. Ovulation can happen anytime. So periodic abstinence is basically you're absent during this particular period. You're not doing sexual intercourse. Now you have one more thing, that is withdrawal method or coitus interruptus, where basically you're withdrawing the penis before ejaculation. That is happening. Now that is very, very risky. Both of these methods are risky, obviously. And there is one more, that is lactational amenorrhea. Lactational amenorrhea means when the baby is being born and the mother is lactating, it seems like ovulation is not going to happen. Why? Because in the mother's body, another hormone is dominating, that is prolactin. Prolactin is the one that releases the, that helps in the release of milk out there. So prolactin does not allow FSH and LH to dominate, which is why ovulation does not happen. Okay, ovulation does not happen in that particular case. So it is considered to be safe by people, but it is actually not safe. It is risky. Okay, it is risky. Now over here, surgical methods, you have tubectomy and vasectomy. Tubectomy, my dear children, happens when you are cutting the fallopian tube out there. Vasectomy, so basically that means you are basically cutting the vas difference in the case of males. Over here, you need to remember, spermatogenesis still happens. Spermatogenesis still happens. Semen is still being produced, but semen does not contain sperm. Seminal plasma and everything is being released, but sperm is not there. So semen does not contain sperm. Spermatogenesis is happening over here. Similarly, in the case of females also, ovulation happens, but menstruation also happens. Ovulation happens, menstruation also happens. Ovum is released, but basically it is tied up, so it cannot move around. Menstruation is also happening out there. Always remember that. Now we are going ahead with the next chapter, that is molecular basis of inheritance. So name the methodologies used in the human genome sequencing. These are the new questions that are coming up. Human genome sequencing, you can do two things. My dear children, always understand your DNA has coding regions and non-coding regions. If at all you are just focusing on the coding regions that are going to form the proteins, you call this as express sequence tag. That is, only the genes expressed as RNA, okay, are identified and sequenced. Only the ones that are expressed as RNA are going to identified and sequenced. Now whole genome sequencing is there. You also call it a sequence annotation, where the entire genome, coding and non-coding sequence, are being sequenced. That are the two sequencing methods in human genome sequencing. Now you have structure of nucleosome. Nucleosome is made up of 8 histone protein. You call it histone octamer. And my dear children, one histone protein is outside also. That is H1 histone. That is a separate thing. Now around this you have the DNA wrapping around twice, my dear children. Almost 200 base pairs of DNA right around here. And also remember the histone protein. Why it provides a perfect base for the DNA to wrap? Because, my dear children, the histone protein is made up of two very important things, that is arginine as well as lysine. Arginine as well as lysine. Okay, they are positive things, and DNA is negatively charged, which is why it can move around very easily. Now, which sequences of bases transcribed from DNA are found both in hnRNA and mRNA? So, when transcription happens, my dear children, the first thing that is formed is known as hnRNA. hnRNA will be having exons, that is the coding sequences, and introns, that is intervening sequences that do not code for anything. Now, what do? You want to make the hnRNA convert into mRNA. For that, you need to make it undergo splicing. It cuts off the introns. When it cuts off the introns, my dear children, what will happen? You will only have the exons, and now these exons are going to code for proteins. That means it can undergo translation. So this is mature mRNA. Now what is common between both of them? Both of them will be having exons. So that is a structure that is very common, and it is being done because of splicing. Okay, splicing is the process of removal of introns out there. Please remember, you have three post-transcriptional modification. You have 3 of them. One is splicing, one is capping, where you add 5-methylguanine at the one end, that is known as capping, and you add adenine groups at the tail, that is known as tailing. Okay, it protects the mRNA from being cut off, because now the mRNA is moving out from the nucleus to the cytoplasm for the process of translation. Cool. Now mention the levels where the gene expression can be regulated in the case of eukaryotes. Now in the eukaryotes, my dear children, you have separate areas, unlike prokaryotes, right? So eukaryotes have a nucleus, then you have a cytoplasm and everything. So nucleus is the place where transcription is going to happen. So I can stop the process of transcription. That is transcriptional level of regulation. Now After transcription, processing can be stopped. That is, splicing and everything cannot be done. In that case also, the whole process will be stopped. So processing level also, this can be done. Next, transport of mRNA from the nucleus to cytoplasm. This mRNA has to move after the cytoplasm. That can be stopped. In that way also I can regulate. Now after that, this mRNA undergoes translation. If I can stop the translation, that level also I can stop it. Okay, so that is one very important thing. Now write a note on the role of sigma factor and the rho factor in the transcription. Always remember over here one more thing. In prokaryotes, transcription, translation is a coupled process. It happens together because everything is in the same area. They do not have any nucleus and things like that. Okay. Now write a note on the role of sigma factor and the rho factor in the transcription of prokaryotes. In the prokaryotes, my dear children, RNA polymerase is going to come and do the transcription. It cannot come and join alone to the initiator promoter region. Usually it goes to the promoter region to attach, but it cannot go alone. That's why it needs sigma factor. It needs sigma factor. Now over here, transcription goes on. Now it reaches the terminator sequence and it has to move down. So in that case, it needs a requirement of rho factor. So just imagine it's crying rho. It is crying out there to move out, to terminate. So terminate with the help of rho factor out there. So sigma and rho factor are super important because of the same reason. Now write the differences between euchromatin and heterochromatin and name the virus which was used in Hershey and Chase experiment. Okay, so euchromatin is basically when, so you have the nucleosomes coming together. It looks like a bead on a string-like structure. Now this structure which is being formed is known as chromatin. This chromatin can be condensed like this, you call it heterochromatin, or it can be loose like this, you call it euchromatin. Over here you see spaces of DNA. Now this DNA can undergo transcription, which is why I'll say euchromatin is what? Transcriptionally active segment. But over here heterochromatin do not have any space, they are transcriptionally inactive. Over here they are loose in structure, which means they will be staining light. Over here they are going to stain dark. These are the few differences that you have. That's all. Okay, now the name the virus which was used in Hershey and Chase experiment, it was bacteriophage. It was bacteriophage, my dear children. It is a virus, it is not a bacteria, and it was affecting E. coli. This was the final experiment to prove that DNA is a genetic material. Now list the goals of human genome project. These are the goals of human genome project. Identify all the genes in the human genome, that is 20,000 to 25,000. Determine the complete sequence, okay, and store the information in the database. Improve the tools for retrieval of the data and transfer the data to somewhere else where it is important, and address all the ethical, legal and everything issues out there. Now DNA is genetic material in both prokaryotes and eukaryotes, yet the packaging process in their cells is not the same. Why is it not the same? In the case of prokaryotes, my dear children, in the case of prokaryotes, the DNA is not too big. The size, genomic size is not too big. In the case of eukaryotes, we have 6.6 × 10⁹ base pairs, right? So obviously genome size is very much different, which is why the packaging is going to be different. Also, the DNA is naked in the case of prokaryotes. Do you need to arrange them into a nucleus? Whereas in the case of us, eukaryotes, we have a nucleus, we need to put it inside the nucleus, which is why in the case of prokaryotes, DNA is naked, circular and present in the nucleoid region without histones because of the small genome and absence of a nucleus. In the case of eukaryotes, we have a nucleus, we need to fit it into the nucleus, which is why we have histones. Then talk about histones, and that's all. Okay, yes. This is an MQP1 question. Now explain the Griffith's transforming principle experiment. Griffith did the experiment to find what is the genetic materials. What did he do? He took Streptococcus pneumoniae. He had two strains, one S strain and one R strain. S strain has a smooth strain. Why smooth? Because it has an extra layer that is known as, that is made up of polysaccharide. Okay, that is made up of what? A polysaccharide out there. That is super important. That is S strain. Okay, so this polysaccharide which is there, or this extra layer which is there, it causes the organism to die. Okay, it leads to the death of the organism. Why? Because this is infective. This is infective in nature. So live S strain which is there, it is going to kill the organism if at all it is there. And R strain, my is the rough strain. It does not have the extra layer, and basically it protects the mouse out there. So if at all I put the live R strain, it was found that the mice lived. If at all I put the S strain, it was seen that the mouse died, which was quite obvious. Now if I put the heat killed S strain, that means I'm treating the S strain with some heat, and then I'm putting it inside the mice was living. That means heat is going to change something up over here in the S strain. That is why the mice is living. And he was shocked, which is why he mixed this and this. Both of them was not causing any trouble initially, but both of them were mixed. He found that the mouse died, and he was astonished. How come that is possible? So then, my dear children, he also got to understand that in the mice there was live S strain, but he never inserted live S strain. So probably this R strain was transforming into S strain because something is going to come from here to here, and that was what? DNA. But was he able to find it? No. He said that it could be DNA, RNA, and any protein out there. We do not know about it. Then Avery McLeod McCarthy came into the picture, and then went ahead and started explaining this, and then came Hershey and Chase. Okay, now state the arrangement of different genes of lactose operon in E. coli. Describe the role of lactose in regulation of lac operon also. So this is the lac operon, my dear children. You have promoter, and this is an inhibiting gene protein out there. This is promoter, again operator ZYA. Now Z over here codes for beta galactosidase, Y over here permease, and A over here transacetylase. Over here this particular protein is going to code, this particular gene is going to code for repressor protein. Repressor protein comes and binds on the operator region, does not allow the transcription to happen smoothly. Now suppose you have lactose or allolactose in the medium, you provide it in the medium, this lactose will come and bind on the repressor protein. This lactose is known as inducer. Okay, now the repressor protein is not allowed to bind over there, which is why transcription happens smoothly. If transcription happens smoothly, the beta galactosidase is going to break down lactose to what? Glucose and galactose. And permease, my dear children, it is going to be broken down. It is not permease, which is there, it is going to help in the increasing the permeability of the cell membrane such that the lactose from outside can come inside. Why are you breaking down lactose? To provide energy. Okay, and transacetylase deals with everything that is in the middle. Okay, so please talk about that. And now given below are the sequences of nucleotides in a particular mRNA and amino acids coded by it, AUG, GGGG, and all of this thing is clear. Write the properties of genetic code. So you have to talk about the properties of genetic code, my dear children. All these properties are mentioned. They are triplet. They are AUG has dual function. One it's code for methionine, other it's a start codon, and some amino acids are coded by more than one codon. You call it degenerate codons. And my dear children, they are read in a contiguous fashion. There is no full stop, comma. There are 3 stop codons: UAA, UA, UGA, and UGA, UAG. Okay, there are 3 stop codons out there, and the codons are universal. That means in the bacteria also it codes the same. In us also it codes the same thing. Now describe the steps involved in DNA fingerprinting technique, my dear children. Isolation of DNA. If I go around explaining, it might be too much. So please go through this. Isolate the DNA, then you cut it with a restriction enzyme, then you separate it, okay, and then you do southern blotting. All the DNA is transferred into a blotting paper. Now the DNA props are being put. The DNA props will be having some it is attached to some what you call fluorescent things. If at all it comes and attaches over here, and if at all you are able to see it under the UV radiation, you will be able to see the bands. Now you can match the band with the band of the same sample that you got from the site of the crime. And then if at all it matches, that means the person has did the crime. Okay. Now concept of transcription unit. This is super important question. Please remember, a transcription unit will be having promoter region and terminator region, 3' over here, 5' over here in this particular sequence. This is known as what? Oh my god, template strand, and this is known as coding strand. Template strand is the one that is going to undergo transcription. And always remember, promoter is going to be like this. Okay, fully covered like this. And the opposite strand of it, my dear children, is the what do you call coding strand, as I told you. Now if at all you want to make the RNA transcribe, please transcribe it and write it on there. The easy way that you can do is write the entire sequence, change the T to U. Change the T to U, because in the RNA there is no thymine, rather it's uracil. Now features of the human genome. These are the features of the human genome. There are 3164.7 million base pairs. The average gene size is this much, and it goes on. Okay. Chromosome 1 has the most genes, that is 2968, and Y has the fewest, that is 231. And scientists have identified about 1.4 million locations where the single base DNA differences have taken place. So these are the important things. This is important. Chromosome 1 with 2968 genes, and chromosome Y with the fewest one, that is 231. The largest gene which is there is dystrophin with 2.4 million bases. The average size is only 300 bases, but over here it's 2.4 million bases. Now explain the process of translation. You have to talk about then. First is activation or charging of tRNA, where the amino acid comes and binds on the tRNA. That is known as charging or activation of tRNA. Okay, and then comes initiation, elongation, as well as termination. Semi-conservative Michelson-Stahl experiment was being performed. It showed that Michelson-Stahl experiment means you have to prove the semi-conservative mode of replication. So that was done. Over here, other method was also there. It was done by Taylor. Okay, on Vishya Faba. Always remember, it was done with using radioactive thymidine. Over here, you used what you call nitrogen. There's different isotopes of nitrogen, N15 and N14. But there it was thymidine that was being used. Okay, and please remember, in eukaryotes we have 3 different types of RNA polymerase. RNA polymerase 1, 2 and 3. RNA polymerase 1 transcribes rRNA, RNA polymerase 2 transcribes hnRNA, and RNA polymerase 3 transcribes tRNA. So 3T, you can remember like that. And RNA polymerase 1 starts with the rRNA itself. That is the primary function. There are other things also which you need to go through. Please go to that. Now, principles of inheritance and variation. Just give me a quick second. Okay, so we are up here So my dear children, recombinants obtained in both Mendelian and Morgan experiment, yet processes involved in different. Comment. So basically in the case of Mendel's cross, he was talking about independent assortment happening up there, and because of which you find more recombinations out there. Independently genes are coming with different genes out there, which is why when they combine, obviously you find new combinations. But in the case of Morgan, he was talking about linkage and recombination. That means basically, I'll try to explain that with the next question also. So over here, suppose you have the chromosomes with A and capital B. Here it is small a and small b, and there is another scenario where there is a here and b over here, and small a here and small b over here. If here exchange is happening, what exactly happens is that you get a new combination, one with capital B with a, small a, and over here you have capital A with small b. You get a new combination altogether. Why? Because the genes are not linked to each other, they are very far from each other. This is what Morgan said. That is how you get new combinations. And over here, when the genes are very close to each other, they are tightly linked. Whenever they get exchanged, they are going to get exchanged together. This will give to lesser amount of recombination out there. That is the difference between linkage and recombination. Linkage is a tendency of genes located close together on the same chromosome to be inherited together. They can be tightly linked or loosely linked like this. If loosely linked, then more combinations would be there. It reduces the chance of new gene combinations and maintains parental character. But recombination, it is a process by which new combinations of genes are formed due to crossing over, due to exchange. It increases the genetic variation by producing new varieties rather than the parental things. Now identify the symbols used in pedigree chart. This means consanguineous mating, that means mating between relatives. Over here it means that male and female bore a child, and over here this child is what? Having a disease. That is why it is being shaded, and it's a male child who's having the disease. Now the next question is, what is haplodiploid sex determination mechanism? Mention an animal that exhibits it. So over here you have male and the female honeybee. You have the male and the female honeybee. Always remember, in the case of honeybees, the female ones which are diploid in nature, the females one will always be diploid in nature. The male one are always going to be haploid in nature. How? So basically, females undergo meiosis to produce gametes with 16 chromosome. Males, since they already have 16 chromosomes only, they will not undergo meiosis rather than mitosis. Now, if they have gametes with 16 chromosome, suppose this and this comes together, you get a female with 32 number of chromosomes. You get a female with what? 32 number of chromosomes. On the other hand, you will have on the other hand, if at all there is a chromosome with only 16 number of chromosomes, they will be turning into a male only, because there is no fertilization happening. Okay, there is a gamete with 16 number of chromosomes will be not undergoing fertilization, and this is known as parthenogenesis, and this is going to form what? The male child out there. Okay, so males are going to be haploid and females are going to be diploid in nature. This is haplodiploid sex determination mechanism. Now define point mutation. Point mutation means it is a single base pair. If at all there are some changes, you call it as point mutation. Example is sickle cell anemia. Instead of having GAG, you have GUG. So instead of coding for glutamic acid, now we are coding for valine. This is known as point mutation. Now the next thing, what are the conclusions drawn by T.H. Morgan from the crossing over experiments in Drosophila? He said that the higher the linkage, that means tightly linked genes will show lesser recombination, and if the linkage is less, they are loosely linked, they will show higher recombination. That was said by Thomas Hunt Morgan. Please remember the numbers. 37.3% recombination is the highest recombination, and 1.3 was the lowest recombination that was being shown. So 1.3 means they are tight. Recombination is less means they are tightly linked. You should understand that they are very close to each other. Now, why did T.H. Morgan select fruit flies? Because they could be grown on simple medium, they could complete their life cycle fast. Single mating could produce so many offsprings out there, and there is a clear differentiation of sexes in between male and the females. Now, what are chromosomal disorders? Chromosomal disorders means number of chromosomes or the structure of the chromosomes is being hampered. For example, Down syndrome, Turner syndrome, Klinefelter syndrome, all of them are chromosomal syndromes. They arise due to aneuploidy, that is loss or gain of a chromosome. Okay, now over here, give reasons for the following. A simple cut results in non-stop bleeding in hemophilia affected individuals, because in hemophilia, very important protein involved in clotting is being affected. Okay, very important protein involved in clotting is being affected, which is why there is a non-stop bleeding happening. Now, Turner syndrome affected females are usually sterile. Why do you think they are sterile in nature? Because in Turner syndrome, you will have a loss of X chromosome. Only one X chromosome is there. So it's the XO condition that is taking place. So Turner syndrome affected females are usually sterile for the same reason. Okay, because they have rudimentary ovaries, they do not have two X chromosomes out there. This is loss of chromosome, you call it monosomy. Okay, monosomy condition. Now in Morgan's dihybrid cross experiment and Drosophila showed that flies having genes for yellow body and white eyes exhibited less recombination. If less recombination is there, that means these two genes are close to each other. They are tightly linked genes. That's why. Inheritance of skin color in human shows different phenotypes. Why do you think humans have different phenotypes? Because over here, my dear children, not only one gene is affecting one particular character, multiple genes are affecting one character. That is why it is an example of polygeny. And your human height is also an example of polygeny. Now, accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why do you think phenylalanine is getting accumulated out there? Because they do not have an enzyme, that is phenylalanine hydroxylase. The gene for that is being mutated. Now this is a autosomal recessive disorder, my dear children, and over here, since there is no enzyme, the conversion does not happen and phenylalanine gets accumulated in the body, okay, in the form of phenylpyruvic acid. Now few autosome linked gene recessive disorders or blood disorders are there. Some are qualitative and other one is quantitative. Now here I would not be going deep, but letting you know, sickle cell anemia is a qualitative disorder because the quality of beta hemoglobin chain is being affected. Okay, quality is being affected, which is why the quality of hemoglobin is affected, which is why the blood cells also changes its shapes off. Okay, similarly in thalassemia, it's a quantitative problem. It's what? It's a quantitative problem. Over there, it's the quantity that is being problematic out over here. Okay, alpha hemoglobin chain might not be produced enough, or beta hemoglobin chain might not be produced enough, which is why it's a quantitative disorder, not a qualitative. Now going ahead, you have defined aneuploidy. Aneuploidy means loss or gain of a chromosome. Give two examples of aneuploidy. What are the two examples of aneuploidy? Down syndrome and Turner syndrome. Down syndrome is trisomy of 21st chromosome. One extra chromosome is there. Turner syndrome is monosomy, where you lose one X chromosome out there. Now karyotype of the individuals affected by Klinefelter syndrome is what? XXY. You have an extra X chromosome. And the symptoms, or basically the features, there will be underdeveloped secondary sexual catus. There is gynecomastia, that is enlargement of the breast in the case of males. Infertility due to lack of spermatogenesis, and tall stature with long arms, learning difficulty in some cases. So these are the things. Overall muscular development will be there in the Klinefelter syndrome. And the karyotype is what? They have 44 autosomes plus XXY, or you also write as 47 XXY. In total, how many chromosomes are there? 47. Usually we have 46, one extra X chromosome is there. This is known as karyotype. Okay, please know the karyotype of Down syndrome as well as Turner syndrome as well. And Turner syndrome, 44 autosomes plus X naught condition, or you write 45 X naught condition. In the case of Down syndrome, they only have how many? They do not have 44 autosomes, instead they have 45 autosomes, and there is XY condition. In that case it will be a male, and 45A plus XX, that will be a female. Okay, yes. Now, represent schematically the result of one gene interaction. You have to talk about tall plants being fused and producing. Please remember the phenotypic and genotypic ratio. Phenotypic ratio is 3:1, 3 tall and 1 dwarf. Genotypic ratio is 1:2:1. Okay, now categorize the following as either autosomal or sex-linked disorders. Hemophilia is a recessive, sex-linked recessive. You have two sex-linked disorders. One is color blindness and the next one is hemophilia. Please remember it. Thalassemia, phenylketonuria, my dear children, they are autosomal linked recessive disorder. Myotonic dystrophy is the only autosomal dominant disorder that is given in your NCERT. Now, what conclusions will you arrive after careful observation of the following given diagram? So, this is the diagram given over here, and you see on the 6th position valine is there. 6th position valine is there. That means what? It is sickle cell anemia, where the mutations has been taken up there. Okay, now identify the disorder by the following symptoms. Individuals with overall muscular development with gynecomastia, it will be what? Klinefelter's syndrome. Now, explain thalassemia as an example of Mendelian disorder. So you have to talk about that. That is autosomal linked recessive blood disorder, and what exactly happens out there. There is alpha globin gene, 2 of them. If at all both of them are affected, then there's a problem. A beta globin gene is there, that they also have 2 alleles out there. Here you have 4 in total, 1 gene, 2 alleles, 1 gene, 2 alleles. So 4 alleles in total. Any of them can affect, depending on the number of them being affected, it will be varying. Okay, similarly over here also you have this. Remember, HBA gene is on chromosome number 16. Here it is on chromosome number 11. Now schematically represent the incomplete dominance by in Snapdragon or Antirrhinum species. Over here red and the white plants were fused, but you got pink plants. Why? Because red was not completely dominant over the white ones. That is why you got pink plants. So the genotypic and phenotypic ratio after the F2 generation is going to be the same. So over here in the case of normal capital T and small t, you would not find the phenotypic ratio to be 1:2:1. You found it to be 3:1. But over here both the genotypic and phenotypic ratio is 1:2:1. Now identify the genetic disorders given below. Sex-linked recessive disorder occurring in about 8% of males and only 0.4% of the females, leading to defect in the cone of the eye. That means males are the ones who are mostly affected, and it's the cone of the eye being affected. That means it is color blindness. Always remember, it is sex-linked recessive disorder. Females can be a carrier, but males will definitely have a disease. Okay, sex-linked recessive disorder affecting only a single protein involved in the clotting of blood. It is hemophilia. Inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine, phenylketonuria. Autosome-linked recessive blood disease, substitution of amino acid in beta globin chain of hemoglobin. Substitution is happening. This is sickle cell anemia. Autosomal recessive linked gene, blood disease, reduced rate of globin chains of hemoglobin. Reduced rate is there. That means you're talking about quantitative disorder, that is thalassemia. Next, represent schematically the results of two gene interaction by taking seed shape and seed color in the pea plant. I have to talk about the entire thing. That is capital R, and I think this is last question, right? So I can explain. Okay, so listen to me carefully. Suppose you have round and yellow seeds and wrinkled and green seeds out there. If at all you fuse, my dear children, first of all you need to understand, think of them as this. So capital R is there, capital Y is there. From this only one can come. So capital R, from here only one can come. It will be capital Y. So whatever you do, these are the gametes that are going to be formed. Similarly over here, small r and small y. When they both fuse, my dear children, in the F1 generation, what will you find? One capital R, one small r, capital Y, one small y. So the chromosomes, they look like this. Capital R, small r, capital Y, small y. This is the parent. Now when you fuse it for the F2 generation, what are the gametes that you're going to get? Listen to me carefully. From here, capital R can come, from here capital Y can come, this is one gamete. From here capital R can come, from here small y can come, this is another gamete. From here small r can come, from here capital Y can come, another gamete. From here small r can come, from here small y can come, another gamete. So you have 4 gametes in total from one particular plant. 4, you put in both the sides and try to fuse it. What will you get? You get a ratio like this, my dear children, that is 9:3:3:1. 16 of them in total, where 9 of them are going to show the dominant trait. What is it? Dominant is round and yellow. Round and yellow. Okay, dominant is round and yellow. One is the recessive trait, that is what? Wrinkled and green. Now 3 and 3 are the new non-parental traits, the new traits. Recombination has taken place over here. So round was with yellow, now round will be with green. Similarly over here, wrinkled was with green, here wrinkled will be with yellow. These are the new things that you can probably see. Got it? I hope you got it. So these are new non-parental character out there. So suppose you are saying that 6 out of, so anyways, so 6 out of 16, my dear children, 6 out of 16 are going to show non-parental character out there. Always remember that. Now, Mendelian disorders may be autosomal or sex-linked, dominant or recessive, homozygous, heterozygous. Anyways, mention the genotypes of the following pedigree symbols. Autosomal dominant. Autosomal dominant, over here the male is being affected. If at all this is an autosomal dominant disease, either in this condition this guy will have it, or in this condition also this guy will have it. You need a dominant thing out there. And this guy is affected also. So this can be this or this. Now, autosomal recessive. Autosomal recessive means the condition should be like this. Then only this person will have it. But over here this female does not have it. This female does not have it. And if you do not want that female to have it, you need to add one male, dominant allele out there. So capital A, same. Okay, got it? Now, sex-linked dominant disorder. So this is a sex-linked dominant disorder. Again, the male is there, but he does not have the effect of it. And it is sex-linked dominant disorder, my dear children. So in that particular condition, he cannot have something like this. He cannot have something like this, because if this came, this is a dominant thing. If this came, he will have the disease. Instead, he should have something like this. In this condition, he would be safe. Recessive one should be there. If a dominant disorder, if we have the allele, then it will be a dominant allele, it will be a problem. So the condition should be at least like this, that is being mentioned out there. Now here, sex-linked recessive disorder. Sex-linked recessive disorder means basically, again over here, the guy should be having something like this, and the female, my dear children, should be having something like this. Both the things would be recessive. See this and this, match both of them. Okay, don't try to find the entire thing. Over here, sex-linked dominant, but you have to find the opposite of it. So please understand that basic thing. Okay. Okay. So it's already 1 hour, 3 minutes. So I think I should be stopping up over here. I can just run you through human health and disease, what is important. Please remember the names of all the causative organisms. Ascariasis is caused by Ascaris. Ringworm is caused by 3 fungi, that is trichophyton, epidermophyton and microsporum. And typhoid is caused by Salmonella typhi. Okay, please remember that. And what are the different ways in which you can find cancers of internal organs? You have computer tomography, you have biopsy, you have MRI, magnetic resonance imaging, my dear children. Describe the 3 properties of cancerous cells: uncontrolled growth, loss of contact inhibition, my dear children, and also metastasis. They move to different areas and they divide up there. And what is a drug which is a depressant that you get from Papaver somniferum? It is heroin. Heroin is a depressant, my dear children. And also, what is the source plant for cannabinoids? It is what? Cannabis sativa. What is the nature of cocaine? What does it cause, my dear children? It causes euphoria and hallucination. Okay, so 3 things are there. What are the lymphoid organs? Bone marrow, thymus, spleen, and you have lymph node, all of it. Bone marrow and thymus are the place where they become antigen sensitive. That means they are formed, they mature, and they become antigen sensitive. They know that they have to be sensitive to certain antigens, but they do not start working up there. Where do they go and show their effect? In the secondary lymphoid organs such as lymph nodes and spleen, and also Peyer's patch and all of it. Okay, please remember that. So effector organ, and this is different. Please go through the schematic representation of replication of retrovirus. The virus brings up this RNA. The RNA is reverse transcribed with the help of reverse transcription to make the DNA. The DNA incorporates into the genome of the human being. The genome will be now, the virus will be utilizing its machinery to produce what? Its own viral proteins and RNA. Remember this. This is super, super important. If at all you have time, please go through it. Okay, please go through the structure of the antibody as well. H2, there are 2 heavy chains, there are 2 light chains as well. This is antigen binding site. Now differentiate between innate and acquired immunity. Innate is there with us from birth, and acquired is basically when you get an infection and things like that. It is non-specific, that is very much specific to antigens. There is no memory being produced here, there is memory being produced there. There are different types of innate immunity, different barriers. What do you call? Cytokine barriers, physical barriers, physiological barriers, and cellular barriers. Please remember all of it. Apart from that, my dear children, one very important thing that was there is okay, if at all you get a snake bite, what type of immunity? Okay, snake bite, and some people give you anti-venom. What type of immunity is something that you are going to receive, my dear children? You receive passive immunization. Ready-made antibodies are given to you. In the anti-venom, you have ready-made antibodies. This is passive immunization. If at all your body is fighting against something and producing antibodies when you get an infection, what is it? Active immunity. Okay, in the case of allergy, which antibody is found the most? Allergy, IgE. From the mother of the milk, what antibody do you get? IgA, A for Amma. Okay, what is the first antibody to transfer from the placenta to the baby? It is IgG. Okay, so always remember, and these are all examples of what? Passive form of immunity, where the mother is giving antibodies, ready-made antibodies to the baby out there. Okay, and filariasis is caused by Wuchereria bancrofti, and my dear children, malaria and all, please go through it. There's a few things that I can probably tell you right now. And this picture is given. You should know, if at all you know the retrovirus reading, then only you will be able to answer it. Otherwise, you will be not. Now, the next chapter that we have is human health and diseases. But as I said, we do not have time, so I will not go to entire thing. I'll just tell you what is important. So please remember, ringworm disease is caused by 3 types of fungi, that is trichophyton, epidermophyton, and microsporum. Ascariasis is caused by what? Ascaris lumbricoides, which is Aschelminthus, and filariasis is caused by Wuchereria bancrofti or Wuchereria malai, and is spread through mosquito bite. Okay, please remember that. There are 3 ways in which you can detect cancer, that is biopsy, histopathology, CT scan, that is computer tomography. With the help of that, you will be able to find if at all there are some internal organs that have tumors or not. MRI is there to find defects in the living tissues as well. But anyway, if at all the question comes, what are the methods through which you can find cancers of internal organs? Write the 3 of them together. Now, what are the features of cancer cell? First, uncontrolled growth. Second one, they have loss of contact inhibition. They will not stop dividing. They will keep on dividing even though they come in contact with other cells. And also metastasis. They will go spread around throughout the body. Now, my dear children, the next thing that we have is the lymphoid organs. Remember, there are two primary lymphoid organs where this lymphoid cells are going to be formed, they're going to mature, and they're going to be antigen sensitive. Okay, they're going to be sensitive to antigens. They don't start fighting up there. Where do they start the fight game? In the spleen, in the lymph node, in the Peyer's patch, in the malt. So that is secondary lymphoid organs where they become effector cells. So effector cells is different from what do you call antigen sensitive, being antigen sensitive. The next thing, very important thing, is the replication of retrovirus. Please go to the picture. They have RNA, they insert the RNA to our cell. In our cell, they use reverse transcriptase enzyme. Their enzyme to convert the RNA to DNA. And now this DNA gets incorporated into our cell, and this DNA, using our machinery, it forms viral proteins as well as viral enzymes and viral RNA and everything out there, and then the virus babies will be moving out. The first cell where it goes in effect is macrophages. It is known as HIV factory, but usually it affects the T helper lymphocytes. So T helper lymphocytes are the ones which are usually being affected. Also remember that the antibody structure, 2 heavy chains it has, 2 light chains it has. This is the antigen binding site. Also remember that. Okay, difference between innate and acquired immunity. Innate is there from us from our birth. For example, our skin, our mucus, everything. So cellular barriers, cytokine barriers, you have this thing, cellular, cytokine, physiological barriers coming under what? Innate immunity. It's present from birth. Acquired immunity you develop after an infection or vaccination. It's non-specific over here. That is very much specific to antigens. There is no memory. Here there will be memory. Always remember that if at all the question is a virus infected cell is releasing something to save the other cells from virus, it is interferons. They come under innate immunity itself. Okay, so please remember that also. Now, apart from that, ascariasis and all, I told you. Okay, why do cancer patients get alpha interferons? What are alpha interferons? They are biological response modifiers. It increases the immunity of the people who are fighting with cancer. Always remember, this interferon is different. Now, name the group of genes that has been identified in normal cells that can lead to cancer. So, in a normal cell, we have proto-oncogenes. When they become activated, you get oncogenes, which basically causes cancer. Okay, so proto-oncogenes, they are problematic. So these are the very, very important things. Apart from that, if someone comes with snake bite, my dear children, snake bite, they are given antivenom. What does this antivenom contain? It contains ready-made antibodies. So this is a type of passive immunization. If ready-made antibodies are given, this is passive immunization. If at all your body is producing the antibodies by themselves, it is active immunity. For example, we are getting an infection, our body produces what? Antibodies. Whenever we get vaccination, we get antigens, weakened antigens, but our body produces antibodies. Vaccination is a form of active immunity. Whereas if at all we are given tetanus injection, or if at all we are given antivenom, that is what you call passive immunization. From mother to child, ready-made antibodies are given, IgA, in the form of milk, through the milk, right? A for Amma, you can remember like that. So there is ready-made antibodies. From the mother, from the placenta to the baby, you get IgG, that is again ready-made antibodies that is given. During allergy, you have IgE, which is increased. Allergy, IgE, please remember that. In allergies, you have histamine production, which causes the overall problem. Histamine leads to overall issues, and to treat it, you have antihistamines, you have adrenaline, and also you have steroids. So these are the few things. Obviously, there are more to it. My dear children, I'll request you to download the PDF that you have been given in the channel, WhatsApp channel. Go through it whenever you have time. I know you might be in bus or somewhere. Just read through the questions out there. It is definitely going to help you out. This is what I could do. I hope this could help you, and I hope the exam goes really, really well, because you guys are awesome, and I am pretty sure you will be doing great. Wishing you all the best, my dear children, and take care of yourselves. Do not be worried. Do not be panicked. Okay? Bye-bye. Take care.